So hi everyone, my name is Ellie Sugimoto and this is Maddux, and today we'll be presenting about the native and invasive algae distribution in Kaneohe Bay in relation to turtle foraging sites.
First, we would like to thank Jack Kay for supporting this fellowship and endowing us with the funding in order to go through with this research, and for all the support given by Iolani throughout this program.
So algae is incredibly important in maintaining healthy ecosystems. Algae are primary producers, which mean they form the foundation of aquatic food systems by converting sunlight and nutrients into organic matter through photosynthesis. And this supports a diverse community of marine life, from microorganisms to larger predators. So how algae is distributed directly affects habitat and resource availability for marine organisms, because algae provide shelter, breeding grounds, and feeding areas for various species, which in turn affects the growth and distribution of other marine organisms, such as coral reefs and seagrasses, for instance. So the changes in algae distribution can lead to shifts in the population of certain types of species. So dominance by a certain algae species may outcompete another, and this can tip the balance and has a tremendous cascading effect throughout the food web. So algae is also super important in nutrient cycling. So they're basically involved in the uptake of nutrients such as nitrogen or phosphorus. So if algae overgrows, and then the oxygen it produces essentially gets trapped in the top of the algae, so that when the algae dies and decays and the animals eat that, they produce carbon dioxide, which gets trapped. And then this ultimately leads to poor water quality and can detriment the ecosystem as a whole.
So algae is a fundamental building block of the ecosystem in Kaneohe Bay. And although Kaneohe Bay does have a tourist aspect of its ecosystem, with people going out to the sandbar and it being filled with people, it is still one of the most biodiverse environments in Hawaii, and if not in the world. Kaneohe Bay is home to hundreds of native algae species, and although they may not be abundant, as evident by our study, they are essential building blocks to the environment and to species that rely on them and rely on their support in the ecosystem, such as green sea turtles, corals, and other foraging species.
So Kaneohe Bay has the most invasive algae on Oahu. So one of the major findings from our research this summer was the high prevalence of invasive algae species at turtle feeding locations in Kaneohe Bay. So although algae is a natural part of the bay's ecosystem, invasive varieties in particular can be incredibly detrimental both to the overall health of the bay and the marine life living within it. Invasive algae such as Gracilaria salicornia and Gorilla ogo were they were introduced to Hawaii's waters in the 1970s. And so the way that they spread is through fragmentation typically, so they can spread asexually very fast. And then this leads to the overgrowth of invasive algae species, which they crowd out native limu and they reduce biodiversity.
So our research question was, how does the distribution of native and invasive algae in Kaneohe Bay correlate with the diet of green sea turtles? So as you can see here, this is a demonstration from a study done by Russell and Balaz that showcased the food diet of green sea turtles in Kaneohe Bay. And with this, we determined which algae that the green sea turtles eat. And as green sea turtles are prone to eating the most abundant algae, we assume that in each of the locations that we sampled, the most abundant algae would be most abundantly consumed by the green sea turtle. And therefore, the algae that we found is most likely the main source of the green sea turtle's diet in each the North, Mid, and South Bay, as you'll come to see.
So the original goal of our research was to determine what the gap is between what turtles eat and where the turtles are located while they are eating in Kaneohe Bay. So to do this, we collected feces samples from 3 different sections of Kaneohe Bay, that being the North, South, and Mid Bay. We extracted and isolated DNA from the feces, and then we amplified the DNA using PCR with an 18S primer. And then, so these primers were conserved at specific regions so that we could have like a broad range of algae when we were extracting and PCRing. So when we sequence them against a reference algae database that we made with the help of Chloe Sonata, who's another Kay fellow. And the goal was to determine exactly what the specific diet of the turtles were.
This is the results from our eDNA analysis. So we found that the DNA that was most frequently detected in the poop was that of human, which led us to believe that it was not turtle feces that we were collecting. Another point of evidence that suggests that it was not turtle feces is that we found no turtle DNA being amplified in our in the barcodes. And yet another piece of evidence as to why it was not turtle was that we found no algae DNA being amplified, which is a main source of the turtle's diet, in which we would have found if it was indeed turtle feces. So yes, the proof that we were sampling and that we collected was human.
So then we switched our project over more to look at the algae side of things. So we gathered data from the North Bay, Mid Bay, and South Bay. So we kayaked out and we laid, and so as you can see on the right-hand side, these are all the individual points or transect locations that we went to, the majority of them. And so they were 200 meters apart, and we just kayaked out and then placed 25-meter transect lines 10 meters apart from each other. And then at random points along each line, we took a picture with a photo quadrant, which we will talk more about.
This is a photo quadrant framer. So we essentially got this from a UH website that detailed the censusing of algae via transects. And what this does is it allows you to take the same photo on the same dimensions of the same photo every single time you take the photo. So we placed a GoPro at the top of the photo transect, and then we simply move the photo transect to 10 different locations along our transect lines, 10 meters apart, in order to gain the 20 photos that we had per transect. And we repeated this for each transect that we sampled. And then we average all of these results in order to get all the algae distribution results gained from the photo quadrant framer, which we'll talk about next, in order to get our distribution of algae in Kaneohe Bay.
So here's an example of one photo quadrant picture where we essentially just divided it into 9 individual squares and then calculated or estimated the average of the type of algae present there and added all the averages up. And so we use this guide to, as you can see on the right-hand side. And as you can see, this is Gracilaria salicornia in the photo quadrant, which is an invasive algae.
So for our results, the first bay that we'll look at is the South Bay. And for the South Bay, we found all invasive species. So the most species that we found were Gracilaria salicornia, which we previously believed we would find. But then we also found cyanobacteria and unknown. So the unknown parts are algae that we cannot identify with our naked eyes, and it was too variable, so that we decided that instead of just guessing and assuming that it was a certain species, that we would put unknown. So yeah, we ended up having 76% Gracilaria salicornia and 22% cyanobacteria, and then 2% unknown.
And the results of the Mid Bay were more split, with Gracilaria salicornia and Dictyspharia being almost equal, and the rest being unknown and either turf algae, Codium medulae, or articulated corallines. And in the North Bay, also the majority of algae was Gracilaria salicornia.
So invasive algae has demonstrated in previous studies to have a higher capacity to absorb nitrogen stored as arginine from wastewater than native algae, as you can see in this study done by Van Houten in 2014. This nitrogen versus nitrogen delta 15 isotope study done by Muscard in 2016, we can see the trend that particulate nitrogen or wastewater signals with nitrogen delta 15, which is a signal that we tested in a previous study last year that we'll get to in a second. But this showcases that wastewater is signaled by the isotope nitrogen delta 15. And with this, we also see that the invasive algae on the left have a higher production of arginine when compared to its total nitrogen mass. So therefore, we can say that wastewater increases the isotope nitrogen in isotope nitrogen delta 15 in algae, and isotope nitrogen delta 15 increases arginine presence in the algae. So native algae has been outcompeted by invasive algae in areas with high nitrogen levels in the past and in previous studies, and with low circulation. So as you saw in the South Bay, we had staggeringly high levels of invasive species. And as you can see here on the map of the South Bay with the tidal circulation, there's almost no tidal circulation in the South Bay, which is primarily due to it being closed off with low current, and also being closed off with sandbar and multiple reefs, which make it so that the nitrogen that gets introduced into the South Bay via wastewater pollution stays in the South Bay, and therefore allows the invasive algae that has a higher capacity to absorb this nitrogen to flourish and to dominate the native algae.
So as Maddux said, essentially the nitrogen delta 15 represents wastewater or human pollution. So as you can see from this graph, there's a correlation between the invasive algae rates and nitrogen or nitrogen levels. So in the mid-bay, as you can see, like the invasive algae and nitrogen levels correlate with each other, and there's stronger prevalence of nitrogen in areas with higher prevalences of invasive algae.
Yeah. So last year I did a study which looked at the link between nitrogen levels in the water represented by the nitrogen delta 15 isotope and rates of pheropapillomatosis in green sea turtles. And as we see here, there is a close relationship between the nitrogen delta 15 levels, which illustrate human pollution, which were ascertained by sampling algae and doing mass spectrometry on algae, and the pheropapillomatosis rates in the same areas of Kaneohe Bay, which were ascertained via censusing. And this showcases a clear relationship between nitrogen delta 15 and pheropapillomatosis in green sea turtles.
And as you can see from the graph on the left, the invasive algae in eutrophied areas have a higher capacity of absorbing nitrogen than their native counterparts. And as you can see from the graph on the right, arginine has been shown to be an indicator of pheropapillomatosis as one of the main amino acids to indicate that.
With this idea that nitrogen in the wastewater is signaled as the nitrogen delta 15 isotope, and that the nitrogen delta 15 isotope has been correlated to arginine in invasive species of algae in the past, we thought it would be good to look at the relationship between the invasive species of algae and pheropapillomatosis rates in the same areas of the bay. And as you see here, there's a similar relationship as there was with the nitrogen delta 15 levels, where in the mid-bay, there's quite low amounts of invasive algae and pheropapillomatosis in green sea turtles. And in the mid-bay, the relationship continues. In the North Bay, the relationship continues, and in the South Bay, the relationship also continues. And this just suggests that the nitrogen delta 15 from the water that is produced by wastewater gets absorbed by the algae, and with that, the algae translates this nitrogen delta 15 and stores it as arginine, which then the turtles consume by eating this invasive algae. And then once the turtles eat this invasive algae, it goes into their system. The arginine goes into their system, and then promotes the tumor-causing herpes virus and pheropapillomatosis.
So in terms of future research, we think that having continuous and long-term monitoring of the algae in certain areas of water is critically important. But as we learned, traditional transect sampling methods are very time-intensive and labor-intensive. So we suggest that in future research, we should use AI to essentially automate like identifying the different types of algae, so that if you have mass amounts of like a lot of pictures, then you can more easily determine what type of algae that is, and it would also be more specific. And so combining the use of AI with traditional methods of transect sampling could provide a more representative way of determining this.
So throughout this entire process, we learned a lot of things. We learned that it is important to communicate with each other and talk about our research and talk about what we're trying to do in the future and our goals. But we also learned that it is important not to be set back by errors and by unforeseen circumstances in your research, such as with the quote unquote turtle feces that we found. And yeah, and with this, we learned that it's just important to pivot yourself and be able to pivot and have backup plans over backup plans so that in the future, when something does go awry, that you'll know what to do and you'll have the ability to come back and still have solid research.
Yeah, and we also just want to remember that the whole purpose of this research is to help the turtles and to help the marine life out there. And in the research process, sometimes it may seem tedious, but in the end, we just want to create a happier environment for everybody. And we also just wanted to thank everybody who helped us, our families for supporting us, Dr. Chan, Mr. Hill, and Mrs. Kobayashi. Thank you so much for all the help you provided in the lab, even when we went through all the setbacks with our false identification. And we really appreciate all the support everybody provided to us.
Thank you guys so much for coming
Thank
out.
you.